Okay, great. And so the third platform project of PERC that will be discussed today is the data management platform. And Dr. Banani Roy will be presenting for this platform. Dr. Roy is an assistant professor in the Department of Computer Science here at the University of Saskatchewan, and she leads the Interactive Software Engineering and Analytics Lab. Dr. Roy is involved with the PERC project, but also with the water security-focused CFREF, the Global Water Features Program, and her group has developed cloud-based data analysis platforms for both of those CFREF projects. Her research areas include big data analytics, software renovation, and interactive software engineering. And her presentation today will explore supporting multidisciplinary data analytics. So please welcome Dr. Roy.
Okay, thank you, Ian, for the nice presentation. Honestly, I'm really glad to be here today in person after the COVID pandemic. It's really nice to see you all. Actually, I made these slides, but Ian already mentioned about me. Just one thing to mention, I work with several graduate students and undergrad students. If anyone is interested in interactive software engineering, please feel free to contact with me. Okay, I have a very short outline. First, I'm going to introduce scientific workflow management system, which actually have been out there for supporting data intensive research. So these are the analytics platforms. Then I'll talk about VSciFlow, which is a visually guided domain specific scientific workflow management system that we have developed as part of the P²IRC data management project. And here I'll talk about how we can use VSciFlow to specify data analysis process easily and in a reusable way using workflows. So it is clear that traditional software system cannot be used for big data analysis. As you can see from the slide, Microsoft Excel blamed for gene study errors, and these errors are severe in genomic data analysis and raised questions on several research findings. In order to overcome the limitations of traditional computation system, scientific workflow management systems are emerging, and different popular scientific workflow management systems are out there, including Galaxy, Cyverse. Have you heard about these platforms? Yeah, that's great. Actually, you should know about these platforms. These are great platforms for data analysis. These platforms provide actually analysis environment and reproducible workflows, and they support the workflows life cycle, which include composition, analysis and execution of workflows. It also supports provenance data management for the workflows, so that you better understand the workflows, why it is working, and you can track the workflows better way. And it provides transparency in your analysis process. So what happens actually in scientific workflow management system, workflows model the analysis process into different steps, as you can see from this figure. And each step is actually associated with a software tool or module or a software application, and which can do a certain task. And each workflow is always assigned with the input data source on which the analysis is performed, and then when the workflow is executed, the input data is transformed into different representations, and the input of one step becomes the output of another step, and it forms a data flow relationship among the different tools. And after execution, a workflow always produces an output that actually gives you good insights about your input data. So here I mentioned a little bit more about scientific workflows, which actually encapsulates different scientific software tools that are written in different programming languages, and provides you the abstraction using which you can connect different software tools with the data flow relationship. And workflows are always reusable by connecting with different data sources, and when you are executing same workflows again and again, or subset of workflows, it produces process data and intermediate data that you can even reuse. Okay? And workflows can be represented graphically. As you can see from this figure, Galaxy is the workflow system that represents workflow graphically. And here the boxes represent the software tool and its configuration, and the links shows the data flow relationship. But workflow can be also represented with textual language. However, the problem is that graphical workflows are good to start with, but when workflow becomes complex, like you can see in this figure, where a lot of tools are involved and a lot of data flow connections are involved, it becomes really messy and difficult for the scientists to handle. But on the other hand, you see the textual workflows that are that supports easy prototyping and scanning, but it is not good for the non-programmer scientist, right? So in order to balance the strengths and shortcomings of both the visual and textual scientific workflows, we have developed GIFS. GIFS is a visually guided, cloud-based scientific workflow management system that actually allows scientists to interact with different software tools, and then make workflows using a domain-specific language, but in a visual guided manner. So how does it work? So here you can see there are 3 panels. In this left panel, you can get access to the different tools that you need for your scientific analysis. And here it shows different data sources that you will use for analyzing. And you can even upload, your data set here. So when, for example, you want to check quality for your genomic sequence, you can drag the tool in this coding editor, and it will automatically actually generate tool, and then with some placeholder data set, then you can drag the data set from here, and this code will be generated almost automatically. And you can keep adding tools by dragging and dropping, and you can make the workflow. And in this way, although you are if you are not a programmer, you should be able to generate some DSL code, and you can compose the workflow within short period of time. And as it has the coding environment, you will get some facilities like commenting code and code completion facilities. And when the workflow is composed, you can run the workflow, and then you can monitor the workflow execution here. And you can run multiple workflows at a time, and you can also execute, like a scheduler is working behind it, but if you want to execute workflow immediately, you can also do that. Then you can see the different steps output in this bottom panel. And the thing is that if there is error, you should be able to see it, and you should be able to see the log here, why certain error is happening. And after composing the workflow, you can save it, and you can make the workflow public, shared, or private based on your wish. And you can again reuse the workflow, change it, but you can even reuse the developed workflow as a sub workflow of other workflow. So there are a lot of flexibilities are provided to make scientists' life easier when working with different kinds of data processing. So here I showed an example. For example, you know that when you are bioinformatician, you always work with the genomic sequence, and you can check the quality of genomic sequence. You can align the sequence with the reference genome, and then you can visualize how the candidate genome is aligned with the reference genome. So if you actually see this workflow in P²IRC, you will actually see this scripted DSL. Okay, but you don't need to write it fully. You just need to little bit tweak it to customize based on your data set, and whether you want to repeat this workflow for multiple data sets, so you can write some kind of while loop. So why we are calling P²IRC is a domain supported language platform, because according to the definition of domain specific language, which offers a minimal set of language features and limited vocabularies. So with P²IRC we have actually this set of language feature, so you can apply condition, and you can apply different like parallelization and some loops, but you will actually interact with domain specific tools that you know for your analysis. That's so, and that when you are interacting with these tools, it will generate the domain specific language code. So that's the main strength of P²IRC. Okay, and tool integration is another important feature of P²IRC. You can interact with existing tools, but if you want, you can add your own domain specific tool that can be written in different programming languages. That's why we are calling that P²IRC is a multidisciplinary data analytics platform, and which allow you to add different kinds of tools from different domains. So we have very good interface for integrating tools in P²IRC, and integrating tools is the most difficult part in scientific workflow management system, because it involves tools written in different programming languages, and tools can be simple but could be also very complex. So we have all these mechanism here. I don't have time to explain, but I will recommend you guys, if you are interested in P²IRC, you should go to the P²IRC manual and check how you can add a custom tool there. So here I have shown some steps for adding tools, and recently we have actually integrated SynMap workflow in P²IRC. It's a bioinformatics workflow working with a bioinformatics professor in our department as a collaborative initiative, and we actually showed that P²IRC is way better than the COSI workflow by integrating a large number of bioinformatics tools, and it actually shows like dot maps, and we have this visualization also available in P²IRC. So we can also integrate different kinds of visualization in P²IRC, so that you don't need to back and forth like a run for different visualization tools for your analysis platform. And this SynMap actually shows the similarities and differences of two different genomes. Yeah, the advantages of using VisciFlow for Koji is that it offers a user-friendly interface with its built-in like a visually guided interaction, and it offers transparency in handling output. Every time you will get a link for each step of your output, then you can visualize the output and you can see how your analysis process is going on, because sometimes it takes a lot of time for running a workflow. So in the middle of the time you might want to see like how much success you have done by executing the workflow at a certain point. VisciFlow actually offers some kind of query language to see that to see the progress of your workflow. And but Cozy doesn't have those support. And also you can use existing tools, but with the different configuration options, you can easily configure your tools. But you can even integrate your own tools if needed. Okay, it makes you a super user kind of thing. Yeah. So actually, a summary student work on this SynMap workflow. We have a poster presentation today. She also won the best presentation award in the summer symposium. It actually shows the acceptability of the VisciFlow in the wider community, because there were 65 summer projects and VisciFlow won the first position in the competition. It's a good thing. And we are also involved with data management project. The thing is that our data management project, we are developing Sci Data Manager, where you can actually put your data. But now we have a VisciFlow platform, which is analytics. But from the data management platform, we can transfer data to VisciFlow. So you can use the data directly from VisciFlow for analysis. And we are also trying to have API so that we can connect with other data management platforms to make the data available to the scientist. Yeah, in future plans, we are integrating data caching, debugging, and features. And these are the people involved in this initiative. So it's a very big project. And I want to acknowledge Dr. Jean for giving us the SynMap workflow. And over the time, I worked with so many researchers at P²IRC Symposium. And here is the conclusion, which is very simple. VisciFlow is the platform that gives you the easy of use interface for composing your workflows. Thank you.